Hello, uh, today I'm discussing diencephalon. Looking at the broad picture of the nervous system, it is composed mainly of three parts: the sensory part that takes stimuli to the center that integrates the information, and the motor part which takes the impulse from the center and is acted upon by the effector organs like muscles or glands. The topic we are discussing today concerns mainly with the sensory input part. If we focus more on sensory input, any sensory tract or almost all sensory tracts is composed of three parts. The first order neuron that takes impulse from uh, the sense organs to the spinal cord. From here, the second order neurons arise which ascend in the spinal cord or in brainstem in the case of cranial nerves, and then this is taken to the thalamus. Relayed in thalamus, and the third order neurons that arise from thalamus take the impulse from here up to the cerebral cortex. So, today what we are discussing concerns thalamus and some other structures related with it, and it is very important to note that thalamus doesn't only act as a relay center for sensory stimuli, but it also plays many other functions which we are going to see in today's discussion. Looking at the developmental point of view, the neural tube, the cranial part of it differentiates into three vesicles: prosencephalon, mesencephalon, and rhomencephalon. Prosencephalon differentiates into the telencephalon and the diencephalon, and diencephalon is the one which we are discussing today. It can further be divided into many parts. So it consists of, the diencephalon consists of paired subcortical neural structures between brainstem and cerebral hemispheres. They form the lateral wall of third ventricle on either side, and functionally it has many diverse roles. Diencephalon can be divided or is divided by hypothalamic sulcus into the dorsal part that consists of thalamus, metathalamus, and epithalamus. Metathalamus consists of medial geniculate body and lateral geniculate body, and epithalamus consists of various structures including the pineal gland and habenular commissure. The ventral part, or the part that lies ventral to the hypothalamic sulcus, consists of the hypothalamus and subthalamus. To specify the location of hypothalamic sulcus, it extends from the interventricular foramen here to the most cranial part of the cerebral aqueduct. So, this sulcus is the hypothalamic sulcus, and we have the ventral part here and dorsal part dorsal to it. This is a hand-drawn diagram showing the location of hypothalamic sulcus between the interventricular foramen here, cerebral aqueduct here, extending between the foramen and the cranial most part of the cerebral aqueduct. Now, we will see in this discussion, we will be discussing mainly thalamus and briefly on the pineal gland, and remaining structures we will see in our further discussions. Thalamus, if we see its functional role, it plays four major roles, and the most important one being as a site for sensory relay. So, all sensory information except olfaction is relayed to cortex by thalamus, and some form of pain is also appreciated at this level. Many motor systems like the cerebellum and basal ganglia, while recircuiting their outputs to the cortex, they relay in the thalamus. It also forms part of Papez circuit that helps to control some emotional and memory information which go to the limbic cortex, the major part of which is formed by cingulate gyrus. And it also have some intrinsic nuclei which are associated with alertness and arousal. So, it also has some vegetative role. Structurally, thalami, they are paired and they are quite large, almost the size of a small egg. It has two ends and four surfaces, and separated from hypothalamus by hypothalamic sulcus as we saw earlier. and the two thalami are connected to each other through the third ventricle by a neural structure known as interthalamic adhesion. Interthalamic adhesion is not present in entire population, and approximately 70% of population have interthalamic adhesion. To further specify the location of the thalamus, we have this transverse section of entire brain. So we have the third ventricle here. We have parts of the lateral ventricles here, and this is the thalamus. Here we have the caudate nucleus, the internal capsule, and the lentiform nucleus. In coronal section, we can see this is corpus callosum, below which this is septum pellucidum. We have two lateral ventricles. We have the midline third ventricle, and the lateral wall of which is formed by the thalamus on either sides. So here we have again the internal capsule, and we have lentiform nucleus here. In sagittal section, which this specimen has been made very beautifully, and we can see the septum pellucidum here, which separates the two lateral ventricles. We have this corpus callosum seen entirely here, and here we see the thalamus. This is the interventricular foramen. This small grayish structure is interthalamic adhesion, and here we have the cerebral aqueduct. So here again we can quite clearly see the location of thalamus. Now summarizing last 3 or 4 slides, now we can say, or we can mention the relations of thalamus on its ends and surfaces. On the ends, we see on the dorsal surface we have lateral ventricle. On the lateral surface we have internal capsule. On medial surface we have third ventricle, and on ventral surface we have subthalamus and hypothalamus, which are parts of the ventral thalamus, ventral and ventral part of diencephalon. So here again, this is the sagittal section where we can see the interventricular foramen and cerebral aqueduct. So we can say at the anterior end lies the interventricular foramen, in the posterior end lies the cranial most part of midbrain. Now if we go inside the thalamus, we see that it is divided into various nuclei by band of white matter, which are called medullary laminae. Now these medullary laminae are of two types: the external medullary laminae, which is here on the lateral side of thalamus, and it separates the reticular nucleus, which is the part of thalamus itself, from the main thalamus. And this lamina, this white matter, contains the fibers which connect cortex with thalamus. It contains reciprocal connections between the thalamus and cortex. The Y-shaped white band which we see here is called internal medullary lamina, and it divides the main thalamic body into various nuclei. Here we have the anterior nucleus, the medial nucleus, and the lateral nucleus. Now this Y-shaped internal medullary lamina contain fibers which connect various thalamic nuclei with each other. So it's quite interesting to note here that the internal medullary lamina connect various nuclei of thalamus with each other, and external medullary lamina connects thalamus with cortex and cortex with thalamus. Now by those white fibers we have seen, it divides the thalamus into various nuclei, and so in the anterior part we have anterior nucleus, medial part we have medial dorsal nucleus and medial ventral nucleus. Similarly on the lateral side we have dorsal triad nucleus, which are further subdivided into lateral dorsal, lateral posterior, and pulvinar. In the ventral triad nuclei, which are divided into ventral anterior, ventral lateral, and ventral posterior. A ventral posterior nucleus is further subdivided into ventral posterolateral and ventral posteromedial nucleus. If we see the same in the diagram, we have anterior nucleus here, we have medial dorsal nucleus here, and if you see previously we had medial dorsal and medial ventral. Now note that medial ventral nucleus is not seen in this particular section. Similarly, here we have two tiers of nucleus, lateral dorsal, lateral posterior and pulvinar in one side, and ventral we have ventral anterior, ventral lateral and ventral posterolateral. Again, if you see here, there were two nuclei, ventral posterolateral and ventral posteromedial, of which the ventral posteromedial cannot be seen from this side. Now, functionally, before we describe in detail the connections of each nucleus, it is very important to understand the general plan of connections, how thalamic nuclei are connected. We have already mentioned earlier, all sensory stimuli except olfaction, they reach cortex by thalamus. So, all stimuli, they go and relay in nucleus, number one. Number two, some motor stimuli, including the ones from basal ganglia or basal nuclei and cerebellum, they reach cortex by thalamus. So, they also relay in thalamus. Now, all nuclei of thalamus, with exception of reticular nucleus, they project to ipsilateral cerebral cortex. And on the other hand, if you see from the cortical point of view, all areas of cerebral cortex, they receive input from ipsilateral thalamus. And similarly, all thalamic nuclei, they receive corticofugal fibers in reciprocal fashion. Now, this means that thalamic nuclei and the areas of cortex, they have bilateral connections. We have both thalamocortical connections as well as corticothalamic connections. Now, this diagram summarizes the individual connections of the nuclei. The anterior nucleus receives efferent from hypothalamus, efferent goes to cingulate gyrus. So, this is basically part of Pape's circuit, which is part of limbic system. So, it affects the emotion, the affective states and memory. Ventral anterior and ventral lateral nuclei receives efferent from basal ganglia and cerebellum. The efferents, they go to motor and premotor cortices. So, basically this is the motor circuit. The ventral posterior nucleus receives efferent from medial lemniscal tracts, the spinothalamic tracts and trigeminal nerve, and efferents go to the primary sensory cortex. So, the thing which we were discussing quite a bit, that all sensory stimuli reach cortex by thalamus, this is the main sensory circuit. Now, we have medial geniculate body, a nucleus, and lateral geniculate nucleus. Medial geniculate nucleus receives efferent from the inferior colliculus, which the stimulus reaches here from ear, and the efferents are taken to primary auditory cortex. So, basically this is auditory connection. And lateral geniculate nucleus, which is part of visual pathway, receives efferent from retinal ganglionic cells, and efferents go to primary visual cortex. So, this is part of visual pathway. Now, if we have idea about these many connections, it is quite sufficient for almost all clinical purposes. But if you are interested in details of these connections, I have some tables here which you can refer, but will not be discussing them in detail. And again, this diagram summarizes in slight detail the connections of individual nuclei. If you require it, you may please refer it, but we will not be describing them individually. Now, when the fibers, they pass from thalamus to cortex bilaterally, as we know, thalamus is quite small in size compared to the cortex. It looks as if fibers are radiating from there, and those radiating fibers are called thalamic radiations. So, we have these four fan shaped radiations which pass through the internal capsule to reach the cortex. So, here this is the anterior limb of internal capsule, this is the posterior limb of internal capsule, and these two are retrolentiform part and sublentiform part of internal capsule. And the destinations are mentioned here for each of the radiations. Coming to blood supply, the major part of the arterial supply comes from posterior cerebral artery, and there are two groups of branches. Posteromedial group supply the medial and anterior part, and posterolateral group they supply the posterior and lateral part of thalamus. It also receives some tweaks from posterior communicating artery, anterior choroidal artery, and posterior choroidal artery, and middle cerebral artery, all branches of circle of Willis. We can see in this diagram we have thalamus in the midline, we have globus pallidus and putamen here, which together form the lentiform nucleus. We have caudate nucleus here, and we have amygdala here. We will be discussing about these structures in slight detail when we discuss basal ganglia. Now here let's focus in the blood supply. We have internal cerebral artery, which forms the anterior, we have internal carotid artery, which gives rise to anterior cerebral artery. We have middle cerebral artery here, which connects with the posterior cerebral artery via the posterior communicating artery. Now posterior cerebral artery is the one which gives the major blood supply to the thalamus, with some contribution from other branches of the circle of Willis. Now discussing some clinically important factors. Thalamic syndrome is a vascular disease also known as Desarine-Rossi disease, caused by obstruction or ischemia in the ventral and posterior part of thalamus. The disturbance are mainly in the somatosensory aspect, with contralateral impairment of proprioception, touch, pain and temperature. Sensations are exaggerated, they become painful and perverted and exceptionally disagreeable. There might also be development of spontaneous pain and emotional instability, resulting in spontaneous or forced laughing or crying. Another very rare disease known as fatal familial insomnia is caused by prions, and it affects the mediodorsal and ventral anterior nucleus of thalamus. It causes dementia and many other neurological symptoms, and it is always fatal. Thalamic cauterization is an invasive procedure which is done to provide relief of pain in patients with terminal cancer. Now let's discuss in brief about the pineal gland, which is also part of diencephalon. A pineal gland is quite strange in that though it lies within cranial cavity in close vicinity of the cerebrum itself and other parts like brain stem and diencephalon, it shows features of an endocrine gland, and it is not connected to, or it doesn't have any neural connections with any other part of the brain. Also, it is also known as the pineal gland, also known as epiphysis cerebri or third eye, one of the circumventricular organs that lack blood-brain barrier. It is stalked, cone-shaped, almost 8 by 5 millimeters in size. It projects back and up from splenium of corpus callosum. If we see the microscopic structure, it shows features of endocrine gland. There are septa which extend into the gland from surrounding pia mater and divide the gland into lobules. These septa also carry blood vessels and fine unmyelinated sympathetic fibers with it. Contains plenty of fenestrated capillaries, and there are two types of cells. The main glandular or parenchyma is made up of pinealocytes, and the supporting structure is formed by astrocyte-like glial cells. The stalk contains mainly astrocytes. After puberty, or approximately after 16 years of age, the gland there is deposition of calcareous granules of calcium and magnesium, and which results in a radio opaque shadow in X-ray. So this is how it looks under microscope. After H&E staining, we have these pinealocytes, and the supporting tissue is formed by astrocyte-like glial cells. This is a X-ray frontal view and lateral view, where we can see that the pineal gland has calcified, resulting in this shadow, and this is the midline. So it also acts as landmark for radiologist. And on the lateral side, we can see its location, that it is in almost the posterior-most side of the cranial cavity. Blood supply, it is highly vascular and it doesn't have blood-brain barrier. It's supplied by medial posterior choroidal artery, which is a branch of, again, posterior cerebral artery. The venous blood is drained by the internal cerebral vein or great cerebral vein. Nerve supply, though it lies within cranial cavity, it is neurologically isolated from rest of the brain, and it receives postganglionic sympathetic fibers via a nerve known as nervi coronari. Now, there are some functions of pineal gland. First and foremost, it has some role in circadian rhythm influenced by light, and this is the tract through which the impulse reaches from retina to the pineal gland. It is said to be anti-gonadotropic, and this is one reason why puberty occurs only after certain age. Up to that age, it secretes certain hormone which inhibit the production of gonadotropin releasing hormone. So once the inhibition of this hormone is withdrawn, there is production of this hormone, and that causes puberty to occur. And it is said to be inhibitory to almost all endocrine glands. So that's all we have for now, and if there are any comments, any suggestions for improvements, or is there is anything which I am doing quite well, I will be very pleased to know. Please be in touch, and there will be more sessions like this in anatomy. Thank you very much for patient listening.